Let's take it away.
Okay, cool. Thanks everyone. I will just turn my video over for now to avoid any network delay. Since as Peter said, I'm overseas now. So hi everyone, I'm Melody. Today I'm going to talk about CellXY, which contains machine learning models to predict sex label for single-cell RNA sequencing data. Single-cell RNA sequencing is a technology that has the ability to separate tissues into their individual cell, and we are able to measure gene activity for every gene in every cell. This technology is important because we have been able to get a better understanding of how normal development occurs. Suppose we have performed the experiment and captured a pool of cells. We need to separate this pool into individual single cell and extract the RNA. Because it's such a small amount of RNA, we need to perform a large amount of amplification. This is sequenced, and we will have lots of short-read sequences which we can align and codify. We end up with a table of counts which has genes as rows and cells as columns. The count here indicates the activity of that gene in that cell. Single-cell data is highly sparse. There are a lot of zeros in the data set, which makes it quite different to bulk RNA sequencing data. So one of the typical quality control steps we do for bulk RNA sequencing data is to check the sex information of the sample. Here is a Mendelian plot for a mouse kidney bulk RNA sequencing data. Each point here is a mouse. The ones in red are females and the ones in blue are males. You can see that the largest source of variation captured by dimension 1 is sex, and the genotype is captured in dimension 2. Exist is a gene that is responsible for X activation in females. It's highly expressed in females and not expressed in males. So we can use it to easily distinguish males and females in bulk RNA sequencing data. But due to the sparsity in single-cell data, it's not a strong signal as we see in bulk RNA sequencing data. When we look at the expression of exist for females in single-cell data, it's common to see many cells with zero expression. Here is a TSNE plot for a mouse kidney single-cell data. Each point here is a cell. The cells are colored by sex. We can see that it's not easy to tell males from females. So when I was doing my Europe project, we were motivated by this study where they pull a male and a female mouse together in order to increase their sample size and decrease the cost of the study. And then they try to use genes on the X and Y chromosomes to tell which of the cells were females and which of the cells were males. They developed a simple criteria based on 1 X chromosome gene exists and 6 Y chromosome genes listed here. They classified any cell expressing exist and no Y chromosome genes as female, any cell expressing 1 chromosome genes and no exist as male, and ignored cells expressing neither or both classes of genes. With this method, they cannot classify 38% of the data, and their sex labels were not even for male and female cells. So we need to build something more sophisticated than that. We decided to use machine learning techniques to solve this problem. Our method consists of 4 parts. We used 1 human PBMC dataset and 1 mouse kidney dataset. Each dataset contains 2 sex labels for each cell. We first pre-process the data, basically remove the low-quality cells and normalize the dataset. And then we needed to decide what features we will use to predict the sex labels. Then we test some commonly used machine learning models, compare model performance, and build our final model. We built human and mouse models separately. I won't have the time to show you everything here, but I will cover some interesting things. Now I'll go into a bit more detail about our features. We try to find genes that can build a boundary between male and female cells. However, because single-cell data is highly noisy and the relative significance to sex label is quite variable among different data sets, thus we don't want to totally rely on individual genes. We find 66 X genes and 34 Y genes. These X genes are located in the X chromosome and are reported to escape X activation. These Y genes belong to the male-specific region of the Y chromosome. We define two genes, super X and super Y, which are the summation of the corresponding X genes and the Y genes. Here is the distribution of exist, super X, and super Y for our human PBMC dataset. Females generally have larger values on exist and super X, whereas males tend to have larger super Y value. However, there are some female cells expressing super Y, and some male cells have count on exist. We train the model with 8% of the data and test on the remaining 20%. The figure here shows the model performance on human testing data. We measure the performance in terms of sensitivity, precision, and F1 scores. You can see that all the tested models show competitive F1 scores. However, logistic regression gives more balanced performance for male and female cells. Logistic regression is simple and easy to interpret, thus we use logistic regression as our final model. We repeat the training process to build a model for mouse cells as well. The mouse kidney dataset we use is not a pooled dataset. You can see that there are no clear differences among those tested models. We also selected logistic regression as our final model for mouse cells due to its simplicity. We apply the trained model back to that original motivating database. We can classify the majority of the cells and leave 21 cells as unable to classify. It's noticed that we classify even number of male and female cells, which align to the proportion of male and female mice used in the experiment. Moreover, we test our trained models on several public datasets from the bioconductor package, and the result is summarized here. The average sensitivity and precision for the tested datasets reached 93%. Now, people are using cell hashing protocols to pool samples. So people would favor using those protocols to actually pool samples together to reduce the costs, rather than relying on deconvolving male and female cells. But we saw that another way to extend the work is to actually detect male and female doublets. In single cell protocol, what we are hoping is that we are capturing single cells, but in some cases, there can be 2 or 3 cells. There are algorithms that already exist to detect doublets, but some of them depend on clustering information of the cells. So we thought another type of doublet you could get is a male and female doublet, and they could potentially be from the same cluster. We will be able to find them because they will have a mixed male and female signature. We generate such male and female doublets by adding count from 1 male and 1 female cell. We use the same features, exist, super X, and super Y as we used before. We also know that doublets will have a higher library size, so we add it as our extra feature. You can see that these male and female doublets show a distinct expression pattern on these features compared to the singlets. We build the training data by randomly selecting 1,000 singlets and randomly generating 1,000 doublets. The training steps are similar to what we did for building the sex-label classifier. We used 80% data for training and 20% data for testing. Since we generate these doublets, we repeat the whole process for 10 rounds to get a more general behavior. We test 3 models, and you can see the random forest gives an improved average performance across 10 repeated rounds, which gives about 90% sensitivity and 91% precision. So we use random forest as our final model. When we applied our doublet detector on the mouse heart dataset, we detect 1% male and female doublets. We further applied one popular doublet detection method, SC Doublet Finder, on this dataset. It finds 885 doublets in total. If you compare the detected doublets by these 2 methods, you will see that there are 36 overlap doublets. You can also see that we are adding this extra 67 doublets that the SC Doublet Finder is not able to get. In summary, we train the machine learning models to predict the sex label for single-cell RNA sequencing data. And we have models for human and mouse cells separately. Our model reaches about 93% sensitivity and precision across 8 public data sets. The package is available on GitHub, and we aim to submit it to Bioconductor. You can call the sex label classifier and doublet detection model by providing the count matrix and the genotype of your own dataset. Here is our suggested workflow to use CellXY. You can start with your preferred cohort control methods and call our doublet detection model to pick male and female doublets. Then you can pass those singlets to our classified sex model to obtain a sex label for your data. If you want to get the features used in the models, you can call the preprocessed functions. We are writing the paper for CellXY, so if you want to know more details, you can check our paper later. As a future work, we will add a batch option to process blocks of cells in serial. We will also test our models on more datasets. Finally, I want to thank Dr. Belinda Phipson and Professor Alicia Oshlag for their time and effort spent on this project. I also want to thank Europe for providing the initial project opportunity. And thanks for listening. I'm happy to take any questions if you have.
Thank you, Melody. There was a big clap. I don't know if you heard it. Yep. And are there any? Oh, yeah, there's a question here.
Hi. So you said that there was the testing data set contained 1,000 single cells. I was just wondering if there could be any doublets in that testing before you start to make your own doublets.
There shouldn't, because so we actually use a different data set, a different human cell data set, which is only one sample. So they did the sequencing like for each sample separately. So there should only be cells from one individual in that data set. And so I'm supposed to just be single cells there. And then we just generate this by adding the count from 1 male and 1 female. So we're not using any pool data set for the doublet detection part. Yeah, it's just single cells. So there shouldn't be any male and female doublets in that data set. Hopefully it helps.
There's another question from John.
Hi, I'm wondering, so with your XY classification, you've chosen to use features of the Super X and Super Y genes. Are you just doing a straight up sum, or are there any weights applied to your genes?
I just did a simple sum. Yeah, we didn't do any weighting, just a simple sum.
Yeah, because the reason I'm asking is, I guess there are obviously genes that are stronger evidence. So for example, if exists, then you should weight that very heavily in whatever score you have. So maybe you can do some kind of weighting of that with another model, and it will give you a much better performance.
Yeah, probably. The reason why we just do a simple sum, so one of the reasons is that because you are not expecting to see all these X genes in every single data set you will test. So it's not a bit, so we actually start with entropy value of those genes, and then we compare the entropy value of those X genes to our sex label, to our sex labels, and then we just like rank them. You will see it's a bit different among different data sets. So the relative significance of individual X genes, they can be a bit variable for different data sets you have. That's the reason why we just do a simple sum instead of just weighting them, because the weight will be a bit tricky to fix the weight. Yeah.
Okay, thank you.
Thanks.
We might continue. There's one question on Slack, but maybe in the interest of time, I'll just go to the next speaker. So maybe Melody, you can check the BioC Asia 2022 channel, and there's a question for you as well. So thanks again a lot, Melody. And thank you.